Hi everyone. So let me just check this if I'm live there. Okay, I think I'm already live on YouTube, but show yet. Okay, anyway, I'll start and check later again if I'm live. Alright, so let me introduce myself to you all. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Okay, so join with us in Telegram. Also, you can join the channel 9 and 10 biology and the Telegram channel of Unacademy, Icse and Icse books. So this link you can find in the description box. The links of all these channels and so specifically the community link and Unacademy channel you can find. So Bucks Bounty link you can find, and also some of these classes are conducted on the platform which are absolutely free. Okay, so you can comfort of your home. So here as well, so basically you can learn from your favorite educator for free. Am I audible, everyone? Okay, let me just see this. I don't know what is happening. Okay, so you can learn from your favorite educator for free. So there are different educators who will be coming up with different sessions. So you will have access to their sessions. Okay, so different educators are there. So you can engage with the educator via poll. You can also clear your doubt live special class. So we'll be conducting the quiz. Hello, I hope this is fine now. So there was some lag actually. So let me just check if this is still there. Yeah, I think now it is fine, right? Okay. So this is fine, I guess now. Alright, so let's get started. So basically there is a 20% offer going on. So that is with the code VANIYT. So this is my code where you will be getting 20% discount on the subscription. So the duration are like this: 24 months, 18 months, 12 months and 9 months. So usually I recommend you all to go with 12 months of subscription, where the current price is 19,838. Okay, and with this offer, so basically you are going to pay just 16,863, with will be saving will be 2,975. So you have to use the code VANIYT to get 20%. So then only you will be getting this offer price. All right, and other than that, so you can even go for higher months, and if you just want to try how exactly this works, so you can try with 9 months of subscription as well. Okay, so let's get started with the structure of a seed. I think now it is fine. So I hope there is no lag now, or is there a lag yet? Hi Prince, hi Nick, okay, hi Kaship Astro, okay, I think this is fine now, okay. Alright, so let's get started then. So this is about the structure of seeds I'll be discussing. So the monocot seed and the dicot seeds, okay. So seed structure, when we mention, so mainly you can find there is a seed coat, okay, and seed coat is basically nothing but the integument. So after the fertilization occurs, so integuments are the ones which are forming the outer coat, that is the outer seed coat and the inner seed coat, okay. So those are respectively called as the testa and the tegmen, all right. And other than that, you can also find there is a cotyledon that is present, and there is a embryonal axis, okay. So cotyledons, the seed coat covering that seed, so that can be cotyledon or it can even be endosperm, okay. So basically covering that you can find a seed coat, and there is a embryonal axis. So if I have to draw this and show, so basically there is one embryonal axis. Let's say this is the embryonal node where the seeds are attached, okay. So here the attachment of seed is done, okay. And now basically there is a covering. So this covering which is there, so this is completely a cotyledon. So let me just show you this, just a second, okay. So this is going to be a cotyledon now, okay. So where the storage of food material is done, and there is a covering. So covering of what? So there is a covering, so seed coat what we call, so there is a covering. This can be testa and tegmen. So there can be testa and tegmen that is present. Anyway, embryonal axis which is this, so it won't be that bigger. So we can just show it like this. So in that case there is a embryonal axis. So there is a common embryonal node will be there, okay. And with that basically there is a covering. So there is an external covering and an internal covering, okay. So now external covering, there is the outer covering. So let's take this as the inner covering and this as the outer covering, okay. So I'll just label this. So just a second. So now this green part what I have labeled. So this is the outer covering. So what is outer covering called? It is a testa, okay. So testa is outer integument, right. So soon after the fertilization occurs, so there is integuments that you find in the ovule. So those are going to form a testa and a tegmen, which are basically the coverings, okay. So this one is the testa, and now you see there is one more covering here. So this covering is the tegmen, okay. So that tegmen is nothing but the inner integument. So this is gonna be the inner integument, okay. And we'll label other things also. So this one is the cotyledon. So both of these where the nourishment is stored. So these two are cotyledon, okay. And here you can find a common embryonal axis. So I can just change the color for that. So now this is the common embryonal axis where both of the seeds are attached. So this one is the embryonal axis, okay. And this itself, once the germination occurs, so you can see arising from this embryonal axis, so there is a plumule formation, okay, and there is a radicle formation. So the radicle will grow towards the soil, the plumule will grow towards the sun, right. Hi, have to learn tissue is learn, but okay. So tissue, you want me to conduct one session. So maybe in the tomorrow, anyway I have already decided the session. So on tomorrow is which day? So Thursday, Friday. Okay. So Friday I'll take one session. So that is on tissues itself. So I'll just come up with certain tricks so that you can learn these tissues. So that is plant tissues and animal tissues easily. Okay. So I'll make one session on that. Okay. Certain tricks and tips to remember these different types of tissue and their function. So I'll just come up with that session on Friday. Okay, so we'll just do that. So I'll explain, and already I have taken a session on tissue, so you can watch those sessions also. All right, so I'll just explain you that. Okay, so here there is a plumule. Okay, so now you know the plumule will start to grow towards the sun, and this plumule is the one which will give rise to a shoot system. Okay, so that is basically, you know, there is a formation of a shoot, you can see. So that is from the common embryonal axis itself, and arising from this cells there is a root system. So now, you know, let's say here is the root system, so there is a radical development. So I'll just label this one is the plumule. Okay, and this part is gonna be a radical which is developing into a root. So basically you can see branches there, so there is a root development which will be occurring. So this will form the root system later on. So there is a shoot system and root system. So this will arise from the embryonal axis having the tissues for plumule as well as the radical there. Okay, yes, so this is anyway taken from Selena itself. So basically I think here also I have added some Selena part. So basically I teach from Selena itself. So you all refer Selena itself, right? So it's better you all refer Selena itself, because you know the ICSE evaluation is mainly done based on the Selena. Alright. So I have taken the evaluation training for class 10. And anyway, even though you are in class 9, so it's always a good thing that you all refer Selena itself, because all the evaluations when they do in the final exams, so the answers are considered from the Selena. Okay, so it's better you all go with the Selena for class 9 and even for class 10. Okay, so let's get started with this. So basically you can see there is an embryonal axis, so where it is having a tissue for the plumule growth. For the radical growth, the plumule will give rise to a shoot system and the radical will give rise to a root system. Flower, so flower tomorrow. So tomorrow I have taken the bisexual flower itself. Okay, so I'll explain the flower in the tomorrow's class. So you can, I think the session is visible now. I just saw on the channel. So you can just click on that bell icon to get notified whenever the class starts. Okay, so tomorrow anyway I'm taking that session. I think around I think 4:15, not 4:15, 1 second. I have a session at 3 to 4. Okay, so it is 4:15. So it is actually at 4:15 only I'm having a session, so you can just enroll for that. Okay, so let's see this. So there is a structure of a seed. So seed structure consists of all of this. So when exactly does there is a formation of seed? So formation of seed you can see only after the fertilization. So after fertilization means there are a lot of changes that are going to occur. So how exactly cotyledon or endosperm is formed? So that is basically formed from the primary endosperm nucleus. So that is, you know, there is a polar nuclei fusion and the male gamete fusion occurs, right? So product of that is the endosperm there. Okay, so that is a triple fusion product. And other than that, this is seed coat. So that is just the integument is forming the seed coat there. The embryonal axis is after the male and the female gamete form. So there is a zygote formation and there is a embryo formation. So from the embryo itself there is a plumule and there is a radical formation that will occur. Okay, so let's have a look at this. So there is something which I did not say about this. So there is a epicotile and a hypocotyle. So let me just explain you in this picture. Okay, so now this is what, this is the common embryonal axis. Okay, and this is the embryonal node where both of the cotyledons are attached. Okay, so now you know from this particular node and the plumule, so there is this region. So you see this region. So this region can be considered as the epicotile region. So that is between the axis and the plumule. So there is a region. So this can be considered as the epicotile. And now between the same region and this radical, so now this region, what is there? So this can be considered as the hypocotyle region. So that is between the radical and the embryonal axis or the node. So this region, what is it? So that is the hypocotyle, and between this embryonal node and then you can see this one plumule. So that is called as the epicotyle. Okay, so let's have a look at this. So here also they have taken a dicot seed itself. So there is an embryo, there are cotyledon formation, so there is a seed coat formation. So you can see there is a radical that they have shown. So there is a epicotile region that they have shown, hypocotyle region has been shown. Okay, so all that comes under the part of a seed itself. So now seed is attached to the stalk by the hilum. So I'll have to explain you this. So what exactly is hilum and other things? One second. So now you know there is a ovary. Okay, so this part is what? This is the ovary part. Okay, so now inside this ovary, so this is what? This is the ovary part. Okay, and this is placed onto the thalamus itself. So now this is the ovary part, and on top basically you can find stigma, style and ovary. So this is going to be ovary now. Okay, and now you know attached to the placenta. So I can just show you a placenta over here. Okay, so let's say this is a placenta tissue. Okay, so attached to this placenta, so this is what? So this one, let's take, there is a placenta. Okay, so attached to this placenta, so there is a formation of the, so you can find there is a ovule formation. Okay, so there is this ovule present. Okay, and this is the common junction of the ovule, where you can see there is the attachment of this embryo and other things. So now you know, so let's say there is the outer integument and there is inner integument. So we can just label this as well. Okay, so now you know this is actually the integument. So this is outer integument. Okay, and so let me just label this. So this is the outer integument, and this part what you see here, so I'll just highlight that. Just a second. Yeah, so now this part what you see now. Okay, so this one yellow part, the labeled one. So yellow one is basically the outer integument. Okay, so one second. So this is gonna be the outer integument. Okay, which will be completely covering this region. Okay, so this is going to be a outer integument, and we'll just label the other one. So there is an inner integument. So this part what you see here, the on the inner side, so this is now a inner integument. Okay, so this part is basically a inner integument. So we can just complete this as such. Alright, so now you know once this is done, so this is the one which is going to form the structure. So this one is inner integument. So this is after the fertilization. So here what is mentioned, the seed is attached to the stalk, that is by the hilum. So now you know this is the stalk. So let me just label this. So this part is stalk, which can even be called as the funicle. Okay, and you see this common junction here where the stalk and the ovule are attached. So this common junction itself can be called as the hilum there. Okay, so hilum is this one. So there is a formation of hilum. So now this whole part, so you see this structure, so there is a placenta and this is the stalk of this ovule. Okay, and now you know this is the ovule structure. So there is a common junction where the ovule and the stalk are attached. So this junction is what we call as the hilum there. Okay, now as it reaches the maturity, the seed is going to lose the water. So seed will lose the water, and at maturity, so this seed is basically about 10 to 15% till be dry. Okay, which will have only about 10 to 15% moisture. Hi Kiran Singh, good evening. Okay, so it reaches seed dormancy. Okay, that is due to metabolic inactivity. So all the enzymes, so here what is mentioned, it reaches the seed dormancy. So enzymes in the seed, so they will remain inactive. Okay, so enzymes in the seeds will be inactive. Okay, and since they are inactive, so seed, we call that the seed is basically in a dormant condition. So that is in a stopped condition where there is no metabolism. So there is no metabolic activities occurring inside the cell, as all the enzymes have remained inactive there. Okay, because the moisture content is not available. So the seed is completely dry, having only up to 10 to 15% moisture, and that's the reason you can find this specific seed to be, one second, yeah, so this specific seed is dry, and since it is dry, so the enzymes are inactive there. So the micropyle is the opening for water and oxygen entry. So let's say there is a seed. Okay, and when you observe a seed, so on the seed, so basically you can observe a scar there. Okay, so just one opening, a scar like structure that you can observe. So that is nothing but the micropyle opening it is. Okay, so through this micropyle, so this is the micropyle opening, and through this micropyle opening, what is going to take entry? So the water will be taking entry. Okay, so that is for seed germination. So this is a seed now. So seed will start to germinate. So that is through the micropyle opening when the water can enter and oxygen exchange can occur. So basically, you know, all the suitable conditions are provided. So basically there is water present, there is oxygen present, and a particular temperature is present. So now you know the seed has got the favorable conditions for the germination, and after getting all the favorable condition, the seed will start to germinate, where there is a formation of plumule and then the radicle, so which in turn will give rise to the shoot system and the root system respectively. Okay, and other than that, so we'll have a look at this. So how exactly fruit formation occurs? So this is cell division in the ovary after the fertilization, so this will form the fruit. So wall of the ovary, so here this is the wall of the ovary, and ovary is the one which is forming the fruit after fertilization. Okay, so this is after fertilization. What happens? So there is a formation of fruit. Okay, that is from the ovary itself. Okay, so now wall of the ovary forms the pericarp, that is the wall of the fruit. So role of the pericarp, when we mention, so it is seed protection and the dispersal. So it need not be like, you know, all the time edible fruit itself. So like we in case, let's see if we are talking about the pea pod. Okay, so pea will be there. Okay, so let's say I'll change the color, just a second. Okay, so let's say there is a pea pod like this. Okay, and attached to the pea pod you can find lot of peas which are there inside. Okay, so there are peas there. So now you know these peas, what you see here on the inner side, so these are attached and these are seeds basically. So this one is now a pea seed. Okay, So we'll just label this to be a pea seed, and this is a fruit, so that is a pea pod it is. Okay, so this one is pea pod. Okay, so now you can see there is a seed and there is a pea pod. So pea pod is nothing but a fruit. So now you know the pericarp is what exactly it is doing. So it is protecting the seed, and also when it opens, so basically you can see a seed dispersal there. So pericarp is having three layers. So there is a epicarp, mesocarp, and there is a endocarp. Okay, so let's have a look at this. So fruit is the enlarged ripened ovary. Okay, so that is the ovarian wall forming the fruit wall, which is basically a pericarp itself. So this itself is the pericarp, which is further divided as the outermost layer, the epicarp, the middle layer as the mesocarp, and the inner layer as the endocarp there, enclosing the seed. So this is taken from Selena again. Alright. So enclosing the seed, so the fruit protects the seed and helps in seed dispersal. So the fruit is basically protected by the seed there. Okay, and sorry, so fruit is basically protecting the seed there, and also it helps in the seed dispersal. So when the fruit opens up, so you can see the seeds are going to disperse, and wherever these seeds fall, so they are going to grow there as a baby plant again, and then they grow to be a other plant. Okay, so examples we can mention. So there is a mango, and another example that they have given is pea pod. So pea pod, as in this is also a fruit itself. So it need not be a edible fruit, but yes, the pea pod is a fruit there, that is the ovary wall, and inside that you can find many seeds. So those are the seeds which are formed there. Okay, and seed, when we mention, so this seed is a ripened ovule, just like we say for a fruit. So it is a ripened ovary. So now the seed is actually a ripened ovule which contains the embryo. So there is a embryo. So embryo will have what? So it will have a tissue for the plumule formation and the radicle formation there. Okay, so in that case, the embryo which develops into a new plant, so the seed coat protects the embryo from mechanical damage, right? And other than that, so there is bean seed and peas are given. So then there is a grain, so as found in maize, wheat, etc. So you can find, or you can actually find, so basically the fruit wall and the seed coat, so they are fused together. Okay, so in this case, seed is different, fruit is different. So you know that the fruit is basically formed from the ovary, seed is formed from the ovule. In case of grain, what has happened? The fruit wall, okay, so the fruit wall has fused. Okay, so this has fused with what? So the seed coat, that is the integument, is also included in that. Okay, so seed coat is fused with the fruit wall, so both of them together, so they are forming the protective layer for the embryo which is developing inside, and that we call as the grain here. So grain is different from the seed. So you should not get confused between seed and a grain. Seed is basically just the ovule, that is the integument alone. Okay, but now when we mention about a grain, so let's say this one is a fruit, and inside this there is a seed. Okay, so now you know the seed wall and the fruit wall, so they both are fused together, so they are forming the protective covering there, and both of these protective covering together, so now you know they are fused, so they are not separate, and that we call as the grain. So there is a difference between seed and a grain, so where the fruit wall and the seed coat are fused. Okay, so we'll go with the other one. So we'll have a look at some of the questions, and then I'll continue with the topic. So the tegmen of seed develops from. So till now I was just discussing about how exactly the seed formation occurs and what structures are forming the seeds there. So now you know the tegmen of seed is developed from what? So is it a perisperm? Is it a funicle? Is it a inner integument or the outer integument there? Yes, anybody? So what Aman is saying, Virat Kohli is feeling sad or what? Okay, so this one is the testa is the outer integument. For the tegmen, it is the inner integument. Okay, so seed may be defined as, so seed is basically, ripened ovule, fertilized ovary, the gynoecium and embryo. So ripened ovule is the seed there. Okay, so then seed is developed ovule, fertilized and developed ovule, developed ovary, the fertilized and developed ovary. So seed, when we mention, so fertilization has to occur and it has to develop itself into a seed there. So it is basically a fertilized and developed ovule. So let's have a look at this one, the dicot embryo. So dicot embryo, till now what we discussed, that is about the formation of seeds like this. So there is a common embryonal axis where the seeds are attached, and now you can find there is a radicle and a plumule as well. So there is a covering, so there is a seed coat which will be there surrounding the seed. So this is basically a dicot seed. Okay, so dicot seed, where you can find the two of the cotyledons there. So this is one cotyledon. Okay, so this is a cotyledon and this is also a cotyledon. Okay, and based on that, so dicot embryo. So विराट कोहली अच्छा नहीं खेलता तो पढ़ाई में मन नहीं लगता. Okay, and that's the reason you are consoling yourself by saying him. Okay, all right. So the dicot embryo has one embryonal axis and two cotyledons. So this is one embryonal axis and two cotyledons you can find. So epicotile is a part of the axis above the level of cotyledons. So what is epicotile? So this is the part of the axis above the level of cotyledon. So let me just label this. So this above the level of cotyledon is what? This is the one. So this is what? This can be considered as the epicotile there. Okay, and then what is given? So this terminates in the plumule or the stem tip. So let's just draw. This is going to terminate where? So this tip of this plumule is basically, this is the plumule, so terminates in plumule or the stem tip. Okay, so hypoctyle is a part of an axis below the level of cotyledon. So this is the cotyledon axis, and now what you see, so this one can be considered as the hypocotyle, which is below the level of cotyledon, and now this terminates in the radicle or the root tip. So let's say this is gonna be a radicle or the root tip there, and this will be terminating, so terminating in radicle or the root tip. So and that's the reason the hypocotyle, when we mention, it is the part of the axis itself. So this is the embryonal axis now. Okay, below the level of cotyledon. So this is the level of cotyledon where both of them are attached, and this can be considered as the hypocotyle. And hypocotyle terminates at what? At the root tip. So this is going to form the root system later on. So root tip is where you can find this hypocotyle will be terminating. Okay, so root tip is covered by a root cap. So now whenever we draw a root, so let's say this is a root now. Okay, so root tip, it is specifically root tip, which will be covered by the root cap. Any idea why there is a root cap present covering this root tip? Hi Adi, so you are here. Nice. So anyway, you have already learnt all this. Yes, any idea why the root tip is covered by the root cap? So always there is a root cap. So let's see, here is the root cap. So it can be like multiple layers of cells you can find. Okay, and basically it is providing a protection. Okay, so this is to provide a protection to the tip of the root, because the tip is having what? The tip is having a meristematic tissue, and that meristem is the one which is helping in the growth of this root. Okay, so if a growth of a root, if you have to see, so that is basically by the continuous cell division itself, so that is by meristematic tissue. So there is a growth, and if the meristematic tissue get damaged while the root grows pushing through the soil, okay, so in that case there is a damage to the cell that can occur, and in order to avoid that, so there is a root cap which will be provided here at the tip of the root. Okay, so the root cap is providing a protection, so that is for the meristematic tissue, so for continuous cell division to occur. So this is the bean seed and is a dicot, and here you can see there is a cotyledon. One second, I'll just label it. Yeah, so this one is the common embryonal axis. You can see epicotile there. So then below this could be the hypocotyle. Then these are the cotyledons. So this is also a cotyledon nerve. Okay, and there is a integument. So there is a testa and tegment. So we can just see this one being a testa and this being the tegment. So there is the testa and the tegment. So these are nothing but the products of the integument which were basically in the ovule. Okay, so that is before the fertilization. All right, just a second. Yeah, so monocot embryo, when we mention, so the monocot embryo is having a single cotyledon, okay, and that is at the end of the embryonal axis. So now axis above the level of cotyledon, so this I have already said, so which is a epicotile and consists of the shoot apex and some leaf primordia. So this is the one. So here you can clearly see. So same way this will be there in the monocot seed as well. So this one is anyway a dicot seed, but for monocot also you can find there is a epicotile region and there is a hypocotile region. So I am confused in plumule where it is present. So I will just show you this diagram. Okay, and there is one, what is the funicle in seed? Okay, so I'll just explain you that as well. So now you know this is the common embryonal node. Okay, so this is the node, and attached to the node you can find both of the cotyledons are attached there. So arising from this common embryonal axis, so you can see there is this plumule arising. So this part is actually called as the epicotile. Okay, so this one is the epicotile, and the tip of this epicotile is basically the plumule, and this plumule is the one which is developing into a shoot system there. Okay, so that is the tip of this epicotile. So we can say between the embryonal node, so this is the node, and then the plumule tip, so you can find a epicotile, and now below here you can find a hypocotyle, so which is extending from the cotyledonary node itself, where it will form the root system. So that is a radicle is going to form a root system there, and between this radicle and the embryonal node, so this region what do you find? So that is a hypocotyle. So plumule, this tip what is there? That is epicotile tip itself is having a meristematic tissue which will form the plumule there, and that plumule is going to form the shoot system. Is that clear? Okay. And what is funicle in seed? So I'll just explain you this. This is funicle. Okay, so stalk is called as the funicle. So now you know this is the ovary. I'll just draw it again and explain. So this one is the ovary. Okay, and attached to the ovary, so you can find certain tissues in there. So that is the placenta. So let me just highlight that. So now you know, let's see, here is the placenta tissue. Okay, so attached to the placenta you can see there is a stalk which is arising. So stalk here is nothing but, so let's see, here is the stalk. Okay, so arising from the placenta there is a stalk, and this stalk is where the ovule is attached. So let's see, here is this ovule formation where you can see this is the micropylar opening, and these are the integuments there. Okay, so this one is the inverted ovule, so just like this it is inverted. Okay, and now you see this is the stalk, so stalk can be called as the funicle, so this one is the funicle. And you see this common junction here, right, where the stalk and the ovule are attached, so this junction which is there, so this is called as the hilum. Yes, igniter, is that clear about the funicle? So funicle is nothing but the stalk of the seed, so that is stalk of the ovule which is present in the ovary, and after the fertilization occurs, so you can see one in pea pod. So let me just show you this. Where did I draw that? One second. Yeah, so you can see this is a fruit now. Okay, and you can see this is a funicle. Okay, so this is the stalk where it is attached over to the placenta. So this tissue over here, so this will all be placenta. So I'll just change the color. So this will all be placenta. So there will be a particular tissue, so that we call as a placenta. And there is a stalk, a thread-like structure, so where it is attached with the seed. So that stalk is the funicle there. So placenta is basically a tissue. Okay, so tissue where a protective support is provided, so that is a tissue inside the ovary. Okay, and that tissue is holding the stalk along with the ovule, that is funicle, so that there is a firm attachment of the seed to the ovary. Okay, so that is by a particular tissue, so where the exchange of materials can also occur, so that you can see in case of human, the placenta, what we talk about, so that is between the mother and between the child. Okay, so that also is a placenta, which is again a tissue itself where the material exchange occur. So here also this tissue is basically holding this ovule. Okay, and that is inside this ovary wall firmly. All right. So this is about the wheat, so the monocot seed. All right, and let's have a look at this. So this is the dicot one, which I already discussed. So this is with respect to bean seed. So let's have a look at this. That is about the starch, I mean the maize grain, where you can find the protective layer. So there is a aileron. Okay, so it is clear. Okay, so let's see this. So now you know the maize seed, when we draw, just a second, I'll just draw and explain. So this is now a maize seed. Okay, and maize seed is also having a slight opening there. All right, and this is having a double layered covering. So this is actually a grain, it is. Okay, so instead we can say this as a grain. And now you know this protective covering which is there, so this is a proteinaceous covering. Okay, so this covering, what we call, so this is a aileron layer. Okay, so this one is what, this is a aileron layer, which is basically made of a protein. All right, so this is made up of a protein. So you can see here also, so there is a aileron layer. Okay, so this one is fused pericarp and then the testum. So here you know there is a proteinaceous layer. So one more covering that you can find, or just beneath this. Okay, so there is a fused integument. Okay, so there is this covering which you can find. Let me just explain you this. Okay, so now you know this part, what is there, the green one what I'm drawing. So this green part is basically, you know, it is a fused fruit wall. Okay, so this one is fused fruit wall. Okay, and if the fruit wall plus what is the seed coat. Okay, so that is integuments are fused there, so seed coat along with the fruit wall are fused. So that is basically, you know, that we call as the fused pericarp. So in that case we can just mention this to be a grain. So it is a maize grain. Okay, and you can find the aileron layer, so it will have a proteinaceous covering also. So now you know where exactly embryo, endometrium, endosperm and all is present. So let's make a partition here. Okay, and just like you observe in this, so there is a endosperm, so there is a epithelial covering. Okay, so this is basically a endospermic covering itself. So this one is basically the endosperm, and here you can find a papery cotyledon. Okay, so this is a monocot seed. The maize, when we mention, it is actually a monocot. So that means one cotyledon will be there. So which is that cotyledon? So this is the papery cotyledon that we talk of, and this papery cotyledon is called as the scutellum. Okay, so this is what? This is scutellum. Okay, so I'll just label this one. So this one is scutellum, which is a papery cotyledon. So why do we say papery cotyledon? Because it is this is a scutellum, and arising from the scutellum, so you can see there is a formation of the radicle, and then there is a plumule. Okay, so now this part, the one which is below over here, so this can be considered as the radicle, and that radicle is covered by a protective covering, so which we call as the coleorhiza. Okay, and now what you see, so this is the plumule which will give rise to a shoot system. So I'll just label this. So this one is the plumule, and this plumule is also covered by a protective covering, so that we call as the coleoptile. Okay, so there is a coleoptile, and this one is coleorhiza. Okay, so this one is coleorhiza. Alright, so these are the protective coverings which are there, and this itself is the embryo actually. Alright. So why walls are fused? So that is a grain, and since there are no tissues in between, okay, so usually what happens between the ovary wall and the integument, so there will be lot of tissues. So certain case, what will happen, there is a degeneration of those tissues, and that's the reason these walls will get fused. Okay, so that is the fruit wall and the seed coat are getting fused, and both of them are together, and that is when we call it as a grain there. Okay, so there is a scutellum which is a cotyledon, so you can find a coleoptile, plumule, radicle, and a coleorhiza. So all of this together, this is basically a embryo itself. Okay, so that is including even the coleoptile, including the coleorhiza. So all of this can be considered as the embryo in a monocot seed. All right, so this is, you can see a seed development after the ovule gets fertilized. Okay, and based on the presence or absence of residual endosperm, so those we classify as albuminous seed and non-albuminous seed. So if there is an endosperm remaining, so let's say the endosperm is still there, okay, and that we call as the albuminous seed. Okay, and if all the endosperm is consumed, okay, so there is no endosperm remaining, which is basically has converted and it has formed the cotyledon, and that we call as the non-albuminous seed. So non-albuminous or albuminous, when we mention, so albuminous seed is still having an endosperm. Okay, whereas in case of non-albuminous seed, complete endosperm, like it is a nutrition. Endosperm is nothing but the nutrition. Okay, so now all this nutritious endosperm which was there, so all that will be used up in forming a cotyledon. So in the cotyledon, the nourishment will be stored now. So complete endosperm is now over, so it is completely converted into cotyledon, and such seeds we call as a non-albuminous, X-albuminous also can be called. Okay, so this one is endospermic. So albuminous is mentioned, so this can also be called as the endospermic seed, and this is non-endospermic or X-endospermic seed also can be called. Okay, so when the endosperm is not there. So difference between coleoptile and coleorhiza. So coleoptile is actually covering the plumule there. So this one is a plumule. So protective covering for the plumule is coleoptile. So you can just remember this is plumule and this is tile. Okay, coleoptile. So both are pronouncing as P. So that is covering the plumule. And here there is a radicle. So covering the radicle you can find this rhiza. Okay, so both are again R, that is a coleorhiza. Again it is a protective covering around the radicle. Okay, so those are basically protecting the plumule and the radicle so that there is a proper growth of root system and the shoot system. Okay, is that clear? Alright, so let's see this. So scutellum is an endosperm of gymnosperm, papery cotyledons of monocots, protective covering of the radicle and protective covering of plumule. So what is scutellum? Which option is right here? B, yes. So this is the papery cotyledon of monocot. So there is only one cotyledon, and that cotyledon is not like all the stronger cotyledons what you see. So that is basically a paper like a very thin cotyledon that you can find inside this maize grain. Okay, so protective layer of radicle during seed germination and endosperm formation is. So protective covering for the radicle is called as what? Coleoptile, coleorhiza, suspensor, plumule. So what is the answer for this? Protective covering for the radicle it is. So it is again a coleorhiza. So it is B option itself. So radicle coleorhiza and plumule it is coleoptile. Yes. So in X albuminous seed, the food is generally stored in. So this is in non-albuminous seed. Where exactly food is stored? Okay. So here I just told you that. So endosperm. So here this is this we call as a albuminous seed. The one where the endosperm is all it is all consumed and the cotyledon has been formed, and that we call as a X-albuminous or a non-albuminous seed, and that is where you can see there is a storage of the nourishment. Yes, so that's right. So it is the cotyledon. Okay, so cotyledon is where the food is stored. So in case of albuminous seeds, the food is stored in dash. Okay, and in non-albuminous seed it is stored in dash. Okay, so in albuminous seed they are asking, and in non-albuminous seed. So already you know about the non-albuminous seed. So what is the option again? So it is again A itself. So in case of albuminous seed, it is the endosperm. Okay, and in case of non-albuminous seed, so it is basically the cotyledon. Right. All right. So we are done with all the questions. So any doubts you have, you can clear your doubts. Okay. And you can also join my Telegram channel, Vani Kari ICSC Class 9 and 10 Biology. Any doubt you have, you can ask me there as well, or on the community as well. So before I end this session, so let me just say about the batches that we have. So there is a Guardians batch, which is an all subject batch, and the start date was July 6th. Yes, you have an option to watch the recordings, and there are many more sessions which are yet to go live, so you can take up the live session. And here the mode of delivery is English, where maths, physics, chemistry and bio will be covered. So if you are subscribing for class 9, so you will be getting class 10 and class 11, 12 subscription absolutely free along with this. So there is a Avengers batch also. Again, this is a all subject batch, where the start date was July 14th. Yes, here also you have an option to watch the recording, and many more sessions which are yet to go live, so you can take the live session. And in this, the mode of delivery is English. So if you are subscribing for class 9, again here also you will be getting class 10, 11, 12 subscription absolutely free with this. And here the maths, physics, chemistry and bio will be covered. Okay, so meet the ICSC achievers. So join an exclusive session with top educators on how toppers strategize their preparation, to score 98.8% in ICSE board exam. So today that is at 7:45 p.m. So you can have look at this session. So basically you can take this session, so you can enroll now. And Manan sir and Arpit Tiwari will be coming live. Okay, so that is on the platform, so you can take up this session and know about the strategies to crack your finals. Okay, so you can also get 20% discount on the subscription, so that is by using the code VANIYT. So this is my code, so with this code you will be getting 20% discount, where the current price is this one and you will be getting this offer price. So with the code VANIYT, and this is the amount that you will be saving. So basically you have different durations, so 24 months, 18 months, 12 months and 9 months. So you can go ahead with any of these. Okay. So or all cotyledon. So basically endosperm itself is forming the cotyledon, and this cotyledon is basically, it is completely cells only. Okay, and it will have a lot of stored food material. So there can be required proteins will be there, so required starch molecules will be there. So there is a storage. So cotyledon is just like a storage which is formed from the endosperm itself, and that will provide a nourishment whenever the seeds will start to germinate. Okay. And why the scutellum is papery? So here in this case the papery scutellum you can find. So that is because the endosperm is on the top. Below that you can find a papery cotyledon. So that is a scutellum. Okay. So now endosperm will pass the nourishment. So there is no requirement of cotyledon here, because all the endosperm is not converted into cotyledon. Okay. So endosperm is only providing the nourishment, and since there is the connection which is there with the cotyledon, always you can find in a seed when we mention. Okay, I'll just draw it here. Okay, so in the seed when you mention, so you can find there is a cotyledon, and with this cotyledon only you can find there is the embryonal axis, which will form a plumule and then the radicle. Okay, and why this is thin is because the endosperm is providing a nourishment, so it will pass the nourishment to the cotyledon, and through the cotyledon the embryo, that is plumule and the radicle, so they are getting the nourishment. So basically through the endosperm it will pass to the scutellum, and then through the scutellum, so since it is attached to the embryo, so through the scutellum itself, so there is a radicle and plumule that are getting the nourishment. Okay. Alright, so introducing community. Okay, so never miss on any update from your favorite educator. So get session notes and practice material. Share your feedback directly with the educator via poll. So you can find the link for the community in the description. So this is opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Alright. So we are done with today's session. I have a session to go on Unacademy platform now. So see you all in the next session. So thank you all. Subscribe to our channel, that is Unacademy ICSE and IC boards. Like and share the video and click on bell icon to get notified about this session, and download the Unacademy Learners app now. So with this PDF if you need, so you can basically join my Telegram, and if you need you can just send me a text there. I'll be sending this PDF on my Telegram itself or on the community. Alright, so see you all in the next session. Bye bye everyone, take care, thank you all. So I have a session to go, so I'll just end this session. Okay, so see you all, bye bye.